Okay. In the first part of this lecture series, we talked about some of the core structure of DNA. We primarily concentrated on nucleic acids and what these nitrogenous bases look like, right? And so we talked about what pyrimidines were, what purines were. We talked about the bases themselves. Now, what I want to do now is to come and talk about the sugars that are involved, particularly with the production of DNA, although we'll also talk briefly about RNA, and then the linker system, which is the phosphoric acid that plays a role. So I want to talk about each one of these, and then what I want to do is come back and put them all together to show you how you get a nucleotide from all three, and then ultimately how we're going to link those together to form these phosphodiester bonds that actually hold the backbone, the chemistry backbone together. So let's look at our sugar component. These are 5 carbon sugars, 1, 2, 3, 4, and then 5 is stuck up on the stem here. Now some people, for whatever reason, try to find this oxygen group right here and call that the 5th carbon because it's part of this pentagon, but the carbons are actually 1, 2, 3, 4, and 5, okay? Now this is ribose. This is not a component of DNA. This is a component of RNA, okay? But what is the component of DNA is a reduced form of this where we're going to deoxy, and we're going to deoxy from the number 2 carbon, which is found right, of course, here. So once we do that, we end up removing this oxygen on the number 2 carbon, and we come number 2 deoxy, okay? Number 2 deoxy over here, and we end up with this different form of ribose. This will be the base unit that we will see as our sugar for the production of DNA. All right, so if we come back, and one of the things that we put on test and things to look for is always whether you've got an OH group hanging off the number 2 carbon or whether you have just a single hydrogen hanging off. You see this form over here, then we're talking about RNA. You see a form where you only have 1 hydrogen, talking about DNA. Now you also will have potentially different groups added onto the number 1 carbon between DNA and RNA because you'll find that there's uracil replacing thymine in RNA, and there'll be thymine replacing uracil, or the proline component here, in DNA, okay? Now, we've got our sugar component. We've got 1, 2, 3, 4, 5th carbon here, 2-deoxyribose, okay? Now, each of the carbon areas have importance in ways. The first one is number 1 carbon is where the base attachment occurs, okay? ATGC. Now, obviously, there won't be a U. U would only occur in ribose, but we're putting that down just to remind you that all these bases attach through the number 1 carbon. Number 2 carbon, now here, is what determines whether you have RNA or DNA, okay, by its form right here, okay? The number 3 carbon and the number 5 carbon become very important then, because these are going to be what's known as the 3', 5' linkage. And lots of genetics problems, we show you a sequence or something, and we say this is 5' end, it's 3' end, which way we're working. Well, that prime ends comes from whether we're talking about an attachment that goes through the number 3 carbon, so off here, or through the number 5 carbon, which is off up here. Number 4 carbon, there's nothing directly attached to it except for this group that's hanging here, that's allowing you to have the 5 carbon off to the side, okay? So that's where our sugar component looks like, this form. Next then, okay, DNA, deoxyribonucleic acid, okay? What makes it an acid is that it's got a form of phosphoric acid is attached. Phosphoric acid is from form that looks like this, phosphate group surrounded by oxygens and incomplete linkage going into those. So you end up with an acid, a negative charge. Okay? So this gives you charge, and in fact will give DNA charge. Okay? So this unit is attached to the number 5 carbon right here by a bond, and that bond is not, that happens to release OH and makes some part of the process of making metabolic water when you do this. But this is known as a phosphoester bond, phosphoester. So phospho with an oxygen in the middle to a carbon and ester bond. Okay, so phosphoester bond, very strong, very important bond that we'll see use. Okay, so this links in through this, making this form. This then brings us back to this 3', 5' discussion. Okay, 3' carbon here, 5' carbon up here on the stalk. Those are what are called our reactive groups, okay? Because you're going to have one of these nitrogenous bases hanging off of this carbon over here. Second carbon is less important for that, and is, you know, other than determining RNA and DNA. But your active groups are here. By active, we mean places where new attachments can be made, right? So that you'll see that the 3' is important because you're going to link 3' down and back to 5' from the other next nucleotide down. You're going to link, I guess you're going to link this 5' up to the 3' going up this way as we build it. Okay, so it's an important unit. Now again, because you're attaching this phosphate group with these negatives over here, you're again making the charge, particularly the outside, it will turn out to be of the DNA molecule, will have a negative charge. That's really important because what's going to happen is that we're going to take this DNA, it's just, you know, in our cells, it's just not naked DNA sticking out there. It's actually wrapped around other proteins. And one of the ways that we have that protein relationship is through electrostatic bonding between this negative charge here, and then the proteins that will be out here that it's attached to will actually be positively charged. So you get a natural attraction. So that's where some of that comes in, okay? Now, I'm a little bit lazy, and what you'll often see me write is something like this, okay? I won't write the whole structure out with all the hydrogens and everything. I'll just write a simple structure that looks maybe something like this. Just be really careful with this. There's the number 1 carbon, number 2, number 3, number 4, and then there's an indication that there's a 5th carbon out here sticking out to this phosphate group, okay? So don't get confused. Again, don't forget that it's a 5 carbon form. All right, so now let's talk about how we actually make a nucleotide. A nucleotide is made up of all 3 components. It's made up of your sugar, your base, your phosphate group, and then of course your nitrogenous base, A, T, G, C, or U, okay? Those are attached in 2 different forms. Pyrimidines are attached with a 1-1 linkage, okay? And again, in biology, as we've said in the other lectures, big name is the smaller structure, smaller name is almost always the larger structure. Okay? So the pyrimidine, which is a single unit, has a 1-1 linkage. A purine has a 1-9 nitrogenous to carbon linkage here. Now, I'm not going to spend a lot of time on this. If you've taken biochemistry, you'll work through every possible bond here and all the different things that are hanging off of this and the hydrogens. I'm just doing a sort of a cartoon version of this because we're interested in not truly the chemistry of DNA. We're interested in how DNA functions as a biological unit, as a genetic unit. So I'm just trying to, you know, kind of bring back to the front of you what generally it looks like. Anyway, these are what you would see as a nucleotide, having all 3 components available. All right, so now we're going to have to link these together. Okay? And in order to do that, we're going to need at least 2, okay, nucleotides. Now, they're not going to be simply in this form, right? When we linkage nucleotides together, they're actually triphosphated. So there would be another one of these phosphate groups out here and another one even further out. So this would be the one closest here. There would be a second and a third. So these are triphosphated. You've all heard of ATP, right? That the ATP is the energy currency of the cell. We use ATP and GTP and all sorts of things for forms. Well, ATP is nothing more than one of these nucleotides with an adenine residue out here that's got 3 phosphate groups. And those phosphate groups are going to be cleaved off to provide the energy that we need to build a bond between this nucleotide here, and this nucleotide up here. And that bond is going to be between this 5 carbon through this phosphate group up through here. So we've got a phosphoester bond right here. We've got a phosphate group, and then we would actually have other energy that we would use to build the form, okay? Now what we would do then is we would use that energy and we would use DNA polymerase, sorry, to help polymerize, to use that energy to help form these bonds. Now we have a bond here between this unit and between this unit. So this carbon through another oxygen, this carbon through an oxygen to phosphate, this phosphate through an oxygen to a carbon. So this is a 5' up to 3' linkage. Okay, 5 carbon up here, all right? And this is the way that it would actually would look, right? Now you have what's known as a phosphodiester bond, this phosphodiester bond. There's one of the ester bonds there. There's another ester bond up here. The carbons, these are very strong bonds, right? And they're very flexible bonds. They're really strong, which because, you know, DNA, you can go out and find DNA from 5,000, 8,000, 10,000-year-old specimens, right? One of the reasons for that is because you've got these phosphodiester bonds that are just so strong that they can tolerate. They can also tolerate, as we go through, we will compress DNA down, we're going to twist DNA, we're going to do all sorts of things. They're very flexible kind of units that allow lots of torquing to go on without tearing up the DNA. Anyway, these are the phosphodiester bonds that are going to be important here. Now, the next phosphodiester bond, the way it's built when we do replication, is to have another nucleotide be down here. A triphosphated nucleotide would come in. We would have DNA polymerase using that energy to form a bond between this 3-carbon right here through the oxygen, down through the phosphate group, and then back down to the nucleotide. So we build going down in this 3' direction. And that'll be important because we'll talk about in replication, which direction do we build in? We'll talk about when we do transcription, which direction do we build in? What that means is which end of this growing molecule do we actually add the next nucleotide to? Okay. All right, so we've got this unit, phosphodiester bond. This is called a dinucleotide. If we know another one on the beer, trinucleotide or a multinucleotide. And again, I'm a bit lazy, so I tend to draw them like this, okay? I tend to indicate where the nitrogenous group is and what it is over here. I shorten the linkage down and it looks something like this. Remember though that there's a carbon in here, 5' carbon. There is a 3-carbon here, and this is a, you know, the 5 to 3 linkage. Directionally though, this is 3', 5'. So you need to learn what that means, okay? Even if I use my shorthand to show something like this. All right, so what have we learned here? What about this, you know, review? It's intended for review. This is all general biology stuff. If you don't know it, like I say in the mini lectures, go back and look at your general biology text because you should know this. I'm simply trying to bring it forward early on in the semester, so when we talk about it, the names mean something to you. Okay, first off is that deoxyribonucleic acid, DNA, is the chemical unit of heritable information. That's what it is, okay, experiments that show that. The structural unit is a nucleotide, which has a negative charge. It's made up of 3 basic components. A sugar, and DNA is made up of deoxyribose sugar. These phosphate groups that help form these phosphodiester bond linkages, and then the nitrogenous bases, A, T, G, or C, right? Those are the base units that make it up. Those are the important units that we would see. All right, so those are the units. That basically concludes this. Now we'll start talking in the next lectures about putting all this together to really make DNA. Okay, because obviously DNA is not just a few little nucleotides strung out. Although RNA tends to be nucleotides just strung out, DNA is a much more complex molecule, and we'll cover that.